Good afternoon, everybody. I'd like to welcome you to our third presentation by Dr. Cesario on the COVID virus. I'd like to introduce you now to Dr. Thomas Cesario. He is the emeriti for the School of Medicine at UC Irvine. And I'm so pleased to introduce him to you. And Dr. Cesario, please take this away.
Thank you, Ellie. I will answer questions, but I think we'll answer all of them at the end of the session and try to do it that way. So let me begin by talking to you about the coronavirus. I'm going to try in the beginning to give you a mini review of a few things that we talked about before, but I like to start by mentioning how we got to where we are. And that is this virus appeared. It appeared in the environment. And it is actually an event that we think has happened before and will happen again. Why is that? Because we think out there are probably something in the order of 3 million different viruses that are capable already of infecting vertebrates. Most of those will never infect humans because they don't have the right attachment device to stick to one of the receptors in our body. And as such, they remain sort of as incidental curiosities to us, at least in the healthcare profession. But occasionally, they do have the right attachment device, or they can acquire it, either because they mutate or they recombine with a different virus, and in so doing, introduce into the population a virus that is now capable of becoming infectious that previously was not. And if that virus is sufficiently unique, we get a pandemic like we have now. If that virus is capable of attaching, entering human cells, and interfering with the function of those cells, in many cases killing those cells, and evoking an immune response. So if those series of events can occur, then you have the potential for a pandemic. And so it is with this virus. We think this virus probably is originally a bat virus that was introduced through an intermediary host. Without boring you with the details, I'm just going to tell you that the virus itself is not unique among viruses, other than for the fact it's new to the human population, but has many of the typical characteristics of a usual virus. And it has 4 major structural proteins. That is to say, 4 building blocks that give it the shape, in essence. Why is that important to us? That's important to us because these proteins are probably the proteins to which our body mounts an immune response, the proteins to which our body will recognize and mount an immune response. And of particular importance to us is the one at the top, we call the spike protein, which is device of this virus. That attachment device is similar to a lot of our other viruses. Those of you who know about influenza know about the hemagglutinin, which is the attachment device for the influenza virus, and so on forth with a lot of the other viruses. We think, and there's, I think, compelling evidence that if you develop an antibody that will attach itself and block that spike protein, you will interrupt the infectious process. And we'll talk about that later on when we get down and discuss vaccines. But we're talking about infections, then a critical question is, what does it take to infect you? And viruses are just like medicines. You have to get the right dose. If you don't get the right dose of the virus, your body can overcome it, inactivate it, or destroy it in some way and prevent it from producing disease. So a critical question to always is, how many viruses do you have to get in order to become infected? And we don't know what this SARS-2 coronavirus, but we think it's probably 100 or so viral particles that have to attach to the receptors in your body, beginning with those in the nose. And I want you to keep in mind that number. How do you get that number? Well, somebody comes along who is infectious, who talks loudly, laughing, coughing, sneezing, and that individual is expelling into the environment a large number of particles of various sizes. Some of them that contain the largest majority terms numerically of viruses, are heavy. They're cased in a lot of liquid, and as a result, they fall to the ground in a short distance and no longer are infectious. On the other hand, some of them are liable to farther distances, and yet some of them are very small. The fluid around them dries out, and as a result, these very small particles, which probably contain a lot less virus, can remain suspended in the air for longer periods of time, and they can travel considerably longer distances. So how that translates is the closer you are to a person who is infectious and expelling virus, the greater the probability that you will get the infectious dose needed to make you sick. This is complicated by the fact that we now recognize that a lot of people are out there who don't realize they're sick, and yet they too are expelling virus into the environment. In the case of families, that number is probably about 17%. What that means is if one family member comes home carrying the virus, living with the others in the house, about on the average, somewhere in the range of 15 to 20% of family members are going to get infected. We also know that we are still continuing to study how many people are asymptomatic and still infectious. There's a brand new study now looking at Marines coming in as recruits, and those studies show that as many as 90% of some of those young men were infectious and not yet actually becoming sick. Furthermore, there are certain events when a lot of people get the virus in a relatively short period of time. We call those events super spreaders. That can happen, particularly if you're in a small, poorly ventilated room and you're in there for a prolonged period of time. Go back to what I told you at the beginning about dose. However long it takes you to get the right dose of viable virus is what's going to allow you to get infected. So here we have examples of choir singers where almost everybody in the choir that night got infected and several people actually died. Multiple events on ships. Down here, one at the bottom is the one in South Korea, which was linked to a church. And there are many, many other events similar to this. In some cases, closed, confined communities like long-term care facilities, that's the LTCF, or food processing plants, where a lot of people were packed together, or prisons. And all these have resulted in many, many cases, sometimes in the hundreds, and as you can see, even in the thousands. But again, going back to what do we need to do to prevent us getting the required number of viruses, that's the protections that we use. Distancing. The farther you are away, the less likely you are to get the required dose. If you put on a mask, that is your final common defense. Put on the mask, and yes, they do trap viruses, and all they're trying to do is keep you from getting the required dose. And there's certainly other things. We know that it's possible on occasion to get the virus on your fingernails or on your fingertips and introduce it into your eyes or your mouth, so we say don't touch. Cough etiquette. We don't want you coughing into the environment if you're infectious. And finally, we want to be able to find early those people who do have the virus, and we want to keep them out of circulation until they are no longer infectious. And for those with mild to moderate disease, that's about 10 days. You are infectious from probably 3 or 4 days before you actually get sick till about 10 days afterward, with the peak of infectivity declining about the 5th day after the onset of symptoms. How do we know that? We know that from studies like those you see here, where we have actually tried to culture live virus from people. And when we do that, we know that in the majority of cases, people with mild to moderate illness are no longer infectious after the 10th day. Yes, there are people with serious illness or who have particular immunologically difficult conditions who can excrete the virus for longer, but in general, it's 10 days and you're no longer infectious. If you do get sick, The major symptoms are fever, cough, and shortness of breath, as you see here, with the usual kind of thing that goes along with a viral illness, which are the aches and pains, the fatigue, etc., that are part of your body's actually immune response to fighting this off. Okay, what happens if you do get sick? Well, we're now learning a lot more about the spectrum of the illness. And probably about 90% of people never really get sick enough to come to the hospital. That doesn't mean they're not sick, doesn't mean they're not in some degree of trouble, but it means that if they stay home for long enough, they will do okay and eventually get over it. Now, getting over it oftentimes means you have a lot of fatigue, a lot of aches and pains, and some cases, other symptoms that can persist for months. But you do get over it and you don't have to come to the hospital. So somewhere in the range of a 1 in 5 to 1 in 10 actually do end up coming to the hospital. And that's what you see here. 75% of those who come for medical attention will have a disease that you can hospitalize for several days, give them fluids, watch their oxygen levels, and ultimately discharge them. But about a quarter of those who get medical attention are going to get sick enough that their oxygen level begins to fall and they end up in the intensive care unit. And of those 25% who got admitted to the hospital, about a third of them will actually require what we call intubation, putting a tube down into the lung and assisting them with their breathing. And about 10% of those in the ICU actually are going to have to put on an artificial lung that we call an extracorporeal membrane oxygenator, or ECMO. And if you get to the stage of being in the ICU and get sick enough, it's not only your lungs that fail, it's oftentimes other organs that are at risk. Your kidneys, for example, your heart, all are at risk of getting damaged when you get to that stage. Your body also does have a way of defending itself. And we know that it begins by mounting an immune response. And that immune response is of two parts. It means you make antibodies and you develop cells that are sensitized to the virus, and through various devices go about ways of arresting the virus. Even if you're asymptomatic, you have an immune response to it. We diagnose this in the hospital or in the clinic, typically by getting a sample from your nose and chemically testing it. We test for the viral RNA. That test tells you whether the virus is there or has been there. It doesn't tell you whether you're infectious. We're not looking for live virus in that circumstance. We know that those tests can be positive for long periods of time, even in the absence of infectivity. We also know that, as I said, when we do the definitive test, which is the viral culture looking for live virus, there are rare people who can have positive viral cultures for long periods, but they are typically people whose immune system is compromised. We can measure your antibodies. We do that clinically, but the test we do in the clinic only tells you whether you have been infected at some point. It doesn't tell you you are immune, and it doesn't do the things that we would like to do, which are to be able to tell you that you don't have to worry about it. It only tells us whether you have been infected. And that brings up another question, and that question is, can you get infected again? And the answer to that appears to be yes. We do know that from the other coronaviruses that repeated infections certainly occur, and in fact, we could say are common. And we now have found at least 26 cases in the world where we can verify and document by carefully done studies that people have gotten the virus at least twice. And in some cases, sick twice, or sick a second time where they weren't very sick the first time. 3 of those have occurred in the US, but as you can see, they have occurred literally all over the world. Really want to talk about today is, where are we with treatment? The usual treatment, which means what you're likely to get if you're sick enough to come to the hospital, basically is modified observation. We give you fluids, we check your oxygen level, We give you nasal oxygen if you need it. And then only if things deteriorate, meaning your oxygen level begins to fall or your blood pressure begins to fall, then we do the more dramatic things. Ultimately, the worst of that is the extracorporeal membrane oxygenator or the artificial lung. Do we have treatments that are definitive? And what I would tell you is we do have treatments, but they are not the optimal. We are not yet at the point where we are confident that we really can treat this and eliminate it before there is any risk. And those treatments fall into three categories: the antiviral agents, the anti-inflammatories, and the immune therapy. So let's look at the one antiviral agent we have, which is remdesivir. Now, this is a very complicated slide, but I just want to show you a couple of things. If you look on the far left, there's a column at the top is in bold. It says recovery. And if you go down to the third line underneath that, it says median time to recovery, and you move your eyes to the right, you will see to the right under the column that says remdesivir, the number 10, and the number that under placebo is 15. That says that the median time to recovery of people who got the drug was 10 days, as opposed to people who didn't get the drug, it was 15 days. And it's on that basis that this drug was approved as usable in the clinic. The time to recovery is faster. But on the other hand, if you go way down to the bottom of that left-hand column that says ordinal score at day, and you look above it and it says Kaplan estimate of mortality, you will see on remdesivir, it says 11.4. And under placebo, just to the right, it says 15.2. That difference is not significant. What does that mean? That says this drug we think may facilitate the rate at which of recovery, but we haven't been able to show that it improves mortality. So we look at this curve for remdesivir. The blue line on that graph is the rate of recovery of people who did not get the drug, and the red line is the people who got the drug. And what I'm trying to show you here is the difference between those who got it and those who didn't was not very great. And in fact, the World Health Organization did a study of over 400 hospitals in 30 different countries, comparing 6 different regimens, including one in which there was no drug at all. And they could not show any difference between this drug and no treatment, or this drug and all the other treatments. So the World Health Organization has gone out on a limb and says they do not support the use of this drug. But our own people here, particularly Infectious Disease Society of America, you'll see IDSA again, say yes, our trial shows faster rate of recovery, so we will use it here. But mind you, the bottom line is nobody has shown that this drug actually impacts mortality. Maybe in the future it will, but so far it does not. What about the anti-inflammatories? Dexamethasone is the synthetic variety of cortisone. It is the most potent cortisone that we have. It's probably 25 to 30 times as potent as cortisone is itself. So the English, in their National Health Service hospitals, of which there are about 175, did a study of over 6,000 patients who got dexamethasone. Now, remind you, when you do these studies, you have a placebo group who got a drug that looks like it, but was basically sugar, and a group that got the drug. So in those studies, what they did is they had 3 results. If you look at the top one, it says that patients who were on mechanical ventilation, those who were in the ICU, we put the tube down and we're giving them the benefit of a breathing machine. That in those cases, when they got to that point, if they got dexamethasone, they had a 30% chance of dying. But if they didn't get dexamethasone, they had a 41% chance of dying. And that difference was significant enough that on that basis, we have said patients who require mechanical ventilation early on should get dexamethasone. On the other hand, what if you do it earlier? If you do it very early and you start using the drug for people who require oxygen, not yet on mechanical ventilation, the difference is 23% died versus 26% died. Still enough of a difference that we say if you are requiring oxygen supplementation because your lungs are that bad or requiring mechanical ventilation, you should get dexamethasone for 10 or more days. On the other hand, if you don't meet those criteria, that is, if you don't need oxygen, or on the other hand, you're already in shock, then probably this drug shows no benefit whatsoever. So it's only in those patients who begin to require oxygen, who go on mechanical ventilation, for which we would use dexamethasone. What about the immune globulin? Well, the first thought was, heck, let's the plasma from somebody who's recovered and give it to somebody who's in trouble. That's convalescent plasma. And well, the anecdotal stories that works, we in fact have never been able to show that it does. So people have said, well, wait a minute. When you are immune, you make a range of antibodies, not just a few, it's a whole family. Let's take the most potent of those antibodies, those that are most able to inactivate the virus, and let's see if we can't produce those synthetically in cell culture and make a population of pure antibodies that are the most potent. And that's what we call monoclonal antibodies. 4 of those have been developed, and probably more at this point, but 4 that we know about, and 2 of those have actually entered clinical trial and usage. The first of those is the Lilly product, which is hard to pronounce, bamlanivimab, and this is a single antibody that's produced synthetically that has gotten to a phase 2 trial, meaning about 500 people got it, half in the control, half in the placebo group. And in that population, as you can see, 465 patients, 5 of the 309 who got it, 2%, actually received the drug and failed and progressed. That is, the majority did not progress. Progress means they got sicker. If they didn't get the drug, 9 out of 156, or 6%, progressed. So the government, our FDA, said based upon that difference, 2% versus 6% progressing to more serious illness, we will approve the drug for emergency use. Now, our own scientists at the Infectious Disease Society and the NIH said, wait a minute, that's not a whole big difference. We want you to use it, but we want that drug investigated farther because we're not convinced that that's really a significant difference. And you haven't even done a phase 3 trial, which I'm going to talk about in a minute. So we use this in the clinic. It's available there, but it's still subject to some controversy. In the meantime, the second company, Regeneron, has their own antibody cocktail. Now, unlike Lilly, which is 1 antibody, this is 2, but being used now on emergency use the same way as the Lilly drug was, and we haven't actually seen their data yet, but the FDA has approved it for emergency use. So what am I trying to tell you? What I'm trying to tell you is, yes, we have made some advances in treatment, which are valuable, but they are not spectacular. We don't yet have a drug that's going to make people who are at death's door suddenly recover and walk out the door. We have just dented the ability to begin to treat this virus, and we are a long way from having a satisfying treatment. So if that's the case, are there other agents that are in the wings? Yes, but they are not yet anywhere near the point that we can say we are excited about them. So if we can't treat, the next question becomes, can we prevent? So we have ways that we can tell you to reduce the dose of the virus, right? Masks, social distancing, quarantine. but we want a more definitive way to prevent the disease. So we develop vaccines. Now, historically, we develop a vaccine is we take the virus, we grow it up, and we treat it. Do that with, or we do it chemically with a thing like propiolactone, but we kill it so that it's no longer infectious, but it maintains its antigenicity. Antigenicity means it maintains its ability to induce antibodies and to induce an immune response. So we can kill it. Or we can grow the virus over and over and over and over again in tissue culture over many passages. And in so doing, the virus, if we're lucky and careful, the virus will lose its ability to make you sick. We call that attenuation. So those are the processes that we have historically used. The whole inactivated virus, that's what we use for influenza. The live attenuated virus, that's what we use for measles, chickenpox, mumps, rubella, etc. So those are examples of what we have used in the past. But now I have said, look, those vaccines on the average took 10 years to verify and certify. In the midst of this pandemic, can we short circuit that process? Are there other things we can do to develop a vaccine that's as good or better? And so we have thought, well, we could just give you the spike protein. Remember, that's the protein that allows the virus to attach. B, we could take the spike protein gene and give you the gene and see if your body will make the protein. C, we could take the gene and add it to another virus and make you think that virus is SARS. And if that virus can't make you sick, but contains the gene and the protein for this particular coronavirus, it will fool your body into thinking it's getting the coronavirus. And you can do that in a variety of ways. So people have sat down and said, well, look, let's use these expedited ways to see if we can't find a vaccine that will be effective and rapidly enough produced that it is useful to immunize billions of people in the world. And so how have we done that? Well, the first thing we do is we take the virus, we see what we can do to modify it, we see what we can do to chop it up into pieces, or we see what we can do to attenuate it. All of those things are being tried. There's, as you'll see in a minute, over 200 vaccine candidates, and they have to go through these stages. First, basic research to develop the vaccine. Then they have to be tried in preclinical studies, which usually means animals. And then they enter the clinical phase. And the clinical phase is of three types. And we'll talk about that just a minute. We say phase 1, phase 2, and phase 3. And then when it comes, the phase 3 is completed, it goes to the Food and Drug Administration for review. And if they like the data, they approve it and release it to be used. But then another committee out of the CDC called the ACIP, which is the Advisory Committee on Immunization Practices, they send a message to the medical community and say, we advise the use of this vaccine or we don't advise the use of the vaccine. Even though it's licensed, we're going to tell you whether we want you to use it or we don't want you to use it. And then you start using it, presumably after everybody approves it, and we start following this to see if there are unusual reactions that we didn't expect or something where the drugs or the vaccine didn't work. So the process has to go through all of these stages, continuously monitored for each of the vaccines. And every time you modify the vaccines, you go through the same process. So what are the clinical phases? Well, the first phase is very small. It's usually about 50 patients. And what you're trying to do, you give the vaccine to those patients and you see, did you get an immune response? Was there any toxicity? Or was it any bad effect on the patient? And if it passes that test, you go on to the phase 2. That's usually about 500 patients. And there again, did you get an immune response? Was it the kind of immune response you wanted? Did you get protective antibody? Did patients get sick with the vaccine? Did they not get sick? And once you have that, then you go on to the confirmatory phase, which is phase 3. Now you're talking about thousands of patients, somewhere between 30 and 60,000 patients are going to be in the vaccine trial. And that's where you're really looking for, does the vaccine prevent disease? And at the same time, you do look to see, are there adverse effects? Did you get an immune response, etc.? But you really want to know from phase 3, did it prevent disease? Okay, so what we said beforehand, that is we, the FDA, the infectious disease community, the research community said, look, before we start approving these vaccines, we want to sit down and say, what is it going to take to convince us that it works? The first thing is evidence of effectiveness. That means in the control group, there were more cases than there were in the group that got the vaccine. The placebo group versus the control versus the vaccine group, more cases got the placebo than got the vaccine. We don't care if you just got sick. We want to know, were there people who really got sick, who had the worst disease? So we want to be sure that there are a difference in the number of severe cases in the placebo or control group versus the vaccine case. Then we want to know, is the vaccine safe? Were there any really bad reactions to it? And then we want a little period of time to go by to make sure that if we miss something, we still could find it, particularly adverse events. So we want a period of a couple months after somebody got the vaccine before we will really say it was safe. So those are the criteria. And it turned out that statistically, to prove the vaccine effectiveness, you had to have about 165 patients who got the disease, and then you would look to see whether they were in the control and whether they were in the vaccine group. You could do a preliminary analysis at about 65 to 70 patients and look the same way, but definitively, you'd have to get to about 165 or 170 patients. So I told you before, there were 200 vaccines candidates that were out there. About 36 of them actually now are in some phase of human testing, phase 1 to phase 3. The ones that we are really interested are down here into phase 3, of which there are 6 that we think are likely. And there are other vaccines in other parts of the world that are at the same stage, a handful, 2 or 3, mainly in China. But those are probably not going to be licensed without being tested in the US. So it's the 6 that we want to look particularly at that are the ones that we're interested in. What are those vaccine types? Well, in some cases, we took just the gene from the virus for that spike protein and gave it to people and said, okay, we're going to give you the gene. If your muscle cells pick up that gene, will they make spike protein to which your body becomes immune? And you can do that either as the RNA, which is the actual RNA of the gene, or you can use it and modify that into DNA, and you could test it that way. So those are the gene-based therapies. The other ones, we said, look, we're going to take a live virus, we're going to modify it so it cannot reproduce in your body, but we're going to cover that virus with the spike protein. So we're going to give it to your body, and your body's going to think it's the coronavirus. It's like putting a wig on the virus, and the wig contains the spike protein. Others have said, well, no, we don't like the idea of giving you a non-replicating virus, because in order to induce an immune response with a non-replicating virus, you have to give large amounts of the virus, which means it's more likely to make you sick. So we're going to give you a virus that can't make you sick, to which we add the spike protein, but which will grow in your body and allow your body to develop an immune response without producing disease. The fourth way is somebody said, well, no, wait a minute. We're all worried about the spike protein. Why don't we just give them the spike protein and see what happens? And we'll show you a vaccine manufacturer that's done that. And then there are finally ways of saying, well, wait a minute, we've been producing the vaccine the old ways for hundreds of years, for 100 years, attenuating the virus by passing it, or by killing. Why can't we try? So those are the types of vaccines that are out there. So the first ones are the genetically based vaccines, of which there are two that are out next week to go to the FDA for emergency use authorization. That doesn't mean approval, but it means they can be distributed and we will begin to use them. Probably the first group will be the frontline doctors seeing these patients. First of those is the Pfizer and the Moderna vaccine. So let's look first at Pfizer, since they released their results first. They enrolled just shy of 45,000 people in their phase 3 trial. Of those, about 40,000 got both doses, the 2 doses of the vaccine, 21 days apart. Now, the problem with RNA is that it's very unstable, and so it has to be kept at a very, very low temperature, which is minus 70 degrees. That's 50 degrees centigrade lower than what than the freezer in your refrigerator has. So it's very, very low temperature. So you say, well, if it has all that trouble, why do we want to use this RNA? Well, it's because A, it's non-infectious. B, you can stabilize it at the cold. It's a very efficient way you can give it to people. It does induce an immune response. And what's most significant is you can make a lot of vaccine very quickly. That's a key here. So Pfizer took the RNA of the spike protein, found a way to inject it into the body, found it got neutralizing antibodies and cellular immunity, and they found that in their final analysis, which was 170 cases, almost all the cases were in the placebo group, 162, and only 8 were in vaccinees. Furthermore, when you look at the seriously ill cases, 11 were in the control or placebo group, 10 in the placebo, only 1 in the vaccine group. So based upon that and the fact there were no serious effects, they submitted the application and is likely to get approved for emergency use next week by the FDA. At the same time, Moderna, which basically did the very same thing that Pfizer in combination with a German company, made their RNA vaccine. Moderna is a US-based company that made their RNA vaccine. They have used it in 30,000 patients, and just like Pfizer, it is the spike protein. It is the gene that makes the spike protein. They have found a way to stabilize it, so you can store this at the normal refrigerator temperature, freezer temperature in your refrigerator, minus 4 degrees. That too, they found it induces a good immune response. They found it was 95% effective. That in their analysis, 95 cases, they found 90 were in the controls, only 5 in the vaccine group, and all the severe cases were in the control group. Now, yesterday they reported, without giving us the actual numbers, they now have the final analysis, and they are saying that this is as good or even better than what they said initially. So based upon that, we think the RNA vaccines will be the first out of the chute, probably will begin to immunize before the end of the month. What about the viral vaccines? Well, the ones that have gotten the farthest are those that are replication defective, meaning you take a virus that's infectious, but when it gets in the body, it cannot reproduce. So the only thing that you give is the virus in the beginning with the mask on it that covers the mask, in this case being the spike protein, to fool the body into thinking it's getting the coronavirus. But once it gets in the body, it cannot divide, and since it cannot divide, it can't produce disease. The only issue with that is, in order to get an immune response, you got to give a lot of this virus, so that more likely you will get fever and some kind of a reaction to it. But these viruses, this type of strategy was tried with Ebola uh, in the case of the Johnson & Johnson vaccine, and actually looked quite good. And a similar thing was tried with Ebola as well, and it looked quite good. So we have two of these that have now come close to being released. The first of those is the Oxford AstraZeneca vaccine. Oxford, meaning that the vaccine was initially developed at Oxford University in England. This is a chimpanzee adenovirus that codes for the spike protein, just like Moderna and just like Pfizer. But it cannot divide in humans. Their phase 3 trial is nearing completion. They had 30,000 people. It looks like their worldwide studies and US studies will probably be completed sometime at the end of December, and likely then they too will go to FDA for approval. They had one case of an unexplained illness, but that always happens in a vaccine trial. It means the trial is temporarily on hold until they certify it's not a serious complication of the vaccine. That was done here. Trial was resumed, and so they are in the final stages of their phase 3 before they apply to the FDA. They have a contract to supply 2 billion doses to the world. Probably, I think this may be the vaccine that is most widely used across the world, partly because you don't have some of the cold storage problems you have with the RNA. Johnson & Johnson has done a similar thing with a different virus. This is not a chimp virus. It's a virus that causes unusual things in humans, kind of minor, causes a little pink eye in humans on rare occasion. It's called adeno 26. They coated it with the spike protein, and so it induces an immune response. Why did they pick this? They picked it because it's a human virus. They thought that would be more likely to give you a good immune response. They did use that for a vaccine used for Ebola in Africa, and it seemed they got good results, but about 5,000 cases. So not a huge experience with it. The good thing is that this virus vaccine is interesting because unlike all of the other things, you may be able to get away with a single dose of this. And that has made it attractive. And they too are winding up the studies. They have shown that they get a good immune response. They also had a brief hold, but they resumed it. They're at finalized phase of the 60,000 patients that they need to do their final analysis. And they too expect to provide 1 billion doses in 2021. Now, all these manufacturers have relied on the fact that they thought they had a product that they were convinced was good enough, they actually could produce the vaccine for distribution before they actually had the final results. It means they had to store it and put it away. Now, Merck, which is a US company, did the same thing using a replicating virus. They took viruses that would not make you sick. They took a cow virus, which is the vesicular stomatitis virus, and they inserted the genes to make that virus make the spike protein, and they are using that. That, when it enters your body, will divide. They're also working on a strategy to take a measles virus that would carry the spike protein and use that potential vaccine. We don't know exactly where they are with that yet, but those are currently in trial. Likely we will see some results in the next year in the Merck vaccine. Finally, we have the protein vaccines. Novavax is a US company based on the East Coast, which has been making vaccines for 20 years, but has never made a successful vaccine in the past. Now, they have been struggling to make a vaccine, but just have not been able to accomplish it. And then coronavirus came along, and they said, wait a minute, we can tell you how to make this vaccine. We've been doing this for 20 years. This is our chance. So what they did is they took the gene for the spike protein and they introduced it into an insect virus, a baculovirus, and they took that virus and infected an insect cell, an army moth cell. And when you do that, the army moth cells produces the spike protein. So you can then scrape it off the cells. We do that chemically, that's not physically. And that protein is harvested. It's mixed with a soap-like particle, which embeds that spike protein, creating what looks like an artificial virus in a sense. And you give that to people, and you get a very good immune response. And in fact, in the preclinical studies, this got the best results. But they've never been head-to-head studies. So when we say in the preclinical studies it was better, it just means that it looked like some of the data was a better immune response, but we actually don't know in a head-to-head trial whether that would be true. They've gone through phase 1 and 2 trials and showing a very good response. They do have to do at two doses, but it's very stable at refrigerator temperature. Again, getting around that issue of how are you going to store and ship this virus. They are in phase 3. They're about to begin phase 3 now. They're going to do it this month, December, and hopefully give us a result probably early next year. So that will be the choices. Choices of the RNA vaccine, the non-replicating viruses, the replicating viruses, the protein viruses. There will be others that are being tried elsewhere. These are the attenuated viruses. There are those that are kill viruses being tested in China. But the front runners here are the ones I've talked about. The RNA viruses, and I didn't mention there's a DNA also that's being tested. And the two viruses, replicating and not replicating, and the protein viruses. So those will be released. Will you be given a choice as to what you're going to get? We don't know. You may very well not be given a choice. The government may say, California gets so much of this, and you'll decide whether you want to take it or not. But on the other hand, it may very well be that we may start out with these vaccines, and the next generation will become available in a year or so, and we'll all get the next generation of vaccines. So that's a moving target. People ask me all the time, will you take the vaccine? And I say, I want to be the first in line because I am ready to try these vaccines. I do think we have enough information that they are good enough, that they will work. We don't know how long they will work for, but I think the important thing is they probably will prevent serious disease in the vast majority of patients. So that brings me to the end of what I wanted to say, and now I'm happy to take questions.
Okay, thank you, Dr. Cesario. We have 3 sources for our questions, and we will take them in the order that we receive them. I will ask the question I received via email. Question is, should someone who has previously had positive COVID-19 and recovered get the vaccination when available?
That's a question we can't answer yet because we don't know what the duration of the immunity is following natural infection. I believe it is likely you will have to get the vaccine at some point because I don't believe the duration of the immunity will be permanent like it is for a disease like measles. I think it's going to be something closer to something like we get with influenza. So yes, I do think you will probably have to do that in the future, but we really don't know the answer to that question yet.
Okay, a question from the Q&A. Our son and his family drove down to visit the in-laws and are quarantining for 2 weeks before they stay with the in-laws. Do they need to quarantine again before coming to stay with us after the in-law visit? So what are your recommendations for all of us with regard to testing?
Yeah, so if you have been exposed to a case, you should be tested. And I would say you should get that test about 5 days after the exposure. Okay, now the second question, if you grew the 14-day incubation period, you don't have to be quarantined again. But what does that 14-day mean? It's 14 days from the day of your last exposure to a case. So if you visited somebody and quarantined for 14 days and they had COVID, it's 14 days after you last saw that person.
Next question is, should someone who has recovered from COVID still take precautionary steps, specifically continue to wear their mask in public?
I would answer that this way. One is that that usually we would expect after the 10-day period of which you are no longer infectious, you would not be worried about infecting others. But I would tell you, yes, wear that mask, because we are still defining people getting a second infection. And we do know you can get a second infection. So even though you've had it once and you're presumably immune, we don't want you to take the risk of getting it twice.
How to properly wear a mask? Could you give us an explanation on that?
Yes. Remember what the mask is going to do. The mask is going to trap virus particles so that it either doesn't escape in the environment or it doesn't get into your nose and mouth, where the receptors for the virus are, particularly in your nose. So you have to cover your nose and you got to cover your mouth. If you don't cover your nose and you don't cover your mouth, you are defeating the purpose of wearing a mask.
From the Q&A, will the vaccines affect our own DNA?
The vaccines will not affect your own DNA. When you give the RNA, which is what Moderna and what Pfizer does, that RNA disappears from the body in looks like 2 or 3 days. It's picked up by the muscle cells. The muscle cells start making the spike protein, but the vaccine itself, the RNA itself disappears in 2 or 3 days. There's no anticipation or any known effect on your own DNA.
And then, can a vaccinated person still be a carrier of COVID?
Can the vaccine person be a carrier of COVID? We don't actually know the question of that to that answer yet, but since you can get reinfected, I would expect it is possible. It may not be as likely, but I would expect it as possible. If you got the vaccine, you will not be a carrier because of the vaccine. You will be a carrier because you get the virus a second time.
How long will a vaccine keep its effectivity?
How durable is the vaccine? Meaning, how long after you get the vaccine are you still protected? Nobody knows the answer to that yet.
From the Q&A, can consumption, for example, eating food including the virus, cause an infection?
No, we know of no relationship between ingestion of food and acquisition of this virus infection. As far as we know, food is safe.
In the initial stages, how do you distinguish between cold or flu and COVID?
You cannot distinguish on clinical grounds. That is, it's not like measles where you can look at the rash and say that's measles. These people get a respiratory infection that looks like influenza, looks like respiratory syncytial virus, human metapneumovirus. The only way you can tell is with a test. And that's what we do the nasal swab for, and we test that way.
From the Q&A, how difficult is it to test for cell-mediated immunity?
The question, how difficult is it to test for cell-mediated immunity? It's not hard to do that. There are a variety of ways that you do that, but it all depends upon how you want to do it. If you want to test specifically to coronavirus cell-mediated immunity, you can do that. You do have to have some special equipment to do that. That's not something that would ordinarily be done in a clinical laboratory. That is something that would be done in sort of a general thing. That is to say, this relates to HIV. When patients get HIV, their cell-mediated immunity declines. So in the clinic, we can do some broad tests that say, is the clinical immunity impaired? But you're not looking specifically at coronavirus impaired immunity or coronavirus immunity per se. You're only looking generally at the cellular immune system. To look at the coronavirus cell-mediated immunity, you can do that, but that is a research tool and only done in research labs working on that.
So from your chart, one of the charts talks about drug therapies, and the chart says that 6% progressed.
Yeah.
Getting worse. Does that mean that the 94% got better?
Yes, that's exactly what it means. It means that those are people who came in with, let's say, mild illness. Yes, it's mild illness. What I didn't tell you on that slide had to do with the immunotherapy, with the Lilly and the Regeneron products. If those products were given to people who were very sick, meaning on the respirator or needing oxygen, it did not work. those immune therapies only work for people with mild disease. So what you're looking for with mild disease, did they progress? Meaning, did they have to go to the emergency room? Did they have to get oxygen therapy? So those numbers told you that of that group, and we're looking particularly now at the Lilly product, that 98% of people who got the Lilly monoclonal antibody did not have to go to the ER or did not have to go into the hospital. On the other hand, if they didn't get it, it was the opposite. 94% had to go in to the emergency room or into the hospital. That's what that difference showed you. Yeah, that's right. That's what progression means. They had to either they had to seek further medical attention.
Question from the chat. What percentage of those receiving the vaccine get a severe reaction?
Let me qualify that and tell you what happens. Probably the majority, more than 50%, will get some reaction. Many of you probably have had Shingrix, which is the shot for shingles. And if you get that, you get a feeling after the second shot. You get a low-grade fever, you feel crummy for a day, you really feel tired, like you should go back to bed. And that lasts a day or two and it goes away. So that's what happens, we think, with the coronavirus vaccines. You'll get a sore arm, like you might get with influenza. You probably get a low-grade fever, 99, maybe 100, 101. You feel kind of crummy, like you're coming down with the flu. And that'll last for maybe 24 or 48 hours and should go away. If you ask for a really bad reaction, what is a bad reaction? That would be somebody who had to really go to bed, had to come into the hospital to be checked, had a fever of 104. And of all the cases with the vaccine, I only know of one case that the fever got that high, and that lasted only a day or two and went away.
From the Q&A, for the phase trials, how do they determine what the sample population will be and who is in it? What age demographics?
For phase 1 and phase 2, I would say first thing is you have to have volunteers. So the first people who are going to be in those trials are people who are willing to volunteer. What does that mean? They're usually young, healthy people, right? So that's probability of what happens. Now, when you move on to phase 2 and phase 3, then you have to start saying, well, we got to not only have young, healthy people, but we better have people who are older, who are debilitated, who have underlying disease, so we know whether it really does work. And then even the question of culture and ethics comes in. You know, in the U.S., so the very important population is, does the African-American population or the Hispanic population respond in the same way that the people of European ancestors or the Asian population, do they respond the same way? So in the big clinical trial, phase 3, you have to have some diversity to show that the vaccine is universally effective.
Are the people in phase 3 actually given COVID-19 to see if the vaccine is working? Are they actually exposed to it?
No, in phase 3, yeah, we don't do those kinds of. The point here is that if you get the vaccine, do we then challenge you with the virus? The answer is no. We give you the vaccine and let you go about your ordinary business, and we see if going about your ordinary business, you happen to, by chance, to encounter the virus. There were people who said we should try that, but was met with a lot of controversy, and it has not been done. So the only thing that's been done with the vaccine is people being allowed to go out of the community, be exposed, and if they get sick, then they are brought in and they are determined whether they have coronavirus or not.
Which vaccine is best for immunocompromised people?
The answer to which vaccine is best for immunocompromised, I don't know. Nobody knows the exact answer to that, but I would say for that type of person, that might be the ideal place where you use the RNA vaccines because they are not infectious. Anything else is infectious, potentially.
Thank you. What will be the priority for vaccines in California?
It's a 4-tier arrangement. And actually, they're meeting today to decide who's going to get it. But the priority that has been reported has been, 1, healthcare workers on the front line. The debate in the phase 1 group is, what about people in long-term care facilities because they have had by far and away the highest mortality rate. And that's being decided now. But mainly it's going to be phase 1 is going to be frontline healthcare workers. And there are at least 20 million doses ready to go, and that basically covers that population. The second phase are the vulnerable. That will be elderly, immunocompromised, people with risk factors. The third group will be otherwise healthy adults. The fourth group will be children. That will be the phases which the vaccine will be distributed.
Does diet have any effect on the virus that we know of?
No, as far as we know, diet does not. Now, diet in general, if you get profoundly nutritionally depleted, that affects your cell-mediated immune system. But you have to be pretty much profoundly nutritionally depleted to that happen. So people who are really, lost enormous amounts of weight and have not been eating adequately for long periods of time, they have a weakened immune system in general. But you have to be pretty severely nutritionally depleted for that to happen.
Thank you, Doctor. Question is, will mask wearing continue to be recommended after general population has been vaccinated?
The answer to that is yes, because there's still a lot of uncertainty about the vaccine. And none of us, I don't think anybody is convinced that these vaccines will be the end to end all, that there probably are better vaccines that will come along in the future. And we still don't know about the duration of immunity. We still don't know about reinfection. And there's still a lot of people out there who will not take the vaccine, who probably are going to be the source of exposure of all the rest of us who did.
It says LA County is now restricting outdoor visits in homes with anyone from outside the household. Do you think this is an overkill? In other words, is it still safe to visit outdoors, socially distanced with a few friends or family members?
I put it in terms like this. Everything that you do carries with it a risk. The question is, how big is the risk that you're taking? If you are socially distanced, wearing a mask, you are outside, the risks are going to be much lower. If you are inside in a poorly ventilated space, close to a person, the risks are going to keep going up. So you have to look at what the circumstances. Remember how I began with dose? The dose of the virus becomes important. Your job is to make sure you don't get that dose. So the farther away that you stand, wearing a mask, being outside, all those are ways of making sure you don't get the right dose. But you always have to put that in context. How big a risk am I going to take? If you go into a bar, it's a crowded bar in a small room, a lot of people in there talking, laughing, then the risks start going up because that virus is aerosolized. Some of that virus is going to remain suspended in the room. And there's a very good chance that if somebody in that room has got the virus, yes, you're going to get the right dose. On the other hand, like say, if you go to somebody outside on somebody's patio, you're distancing yourself and you're wearing a mask, I think the risks are very low. I could tell you that I have friends who I appreciate and think dearly of, who, when they get together, they want to do hugging and kissing and hugging everybody. And that's a recipe for a disaster. So you have to put it in those contexts.
Will post-testing be required for those who are vaccinated as part of measuring efficacy of the vaccine?
Yeah, that's a very good question. And I've not heard a policy statement on, but I think there is a very good possibility that will be the case, that you will have to have your antibodies measured afterward. Or put it this way, you will want to have your antibodies measured afterward.
Being a senior with underlying medical conditions, how do we get on the list for early vaccination?
That hasn't been defined yet. All of those things are being worked out as we talk. I think you'll get a lot of clarification after they get the vaccine approved, which I think will be within the next couple of weeks. And then it will be worked out probably for distribution after the first of the year. Because like I said, the first people to be immunized with the vaccine that's readily available are going to be the hospital workers. And some of that will also probably spill over to the groups like the transportation workers and things like that. But the rest of it, the details, I think will get worked out as we go along, and we find out what the criteria they're going to use and how they're actually going to distribute the vaccine.
Can you comment on distribution of vaccines in states other than California? And what about Midwest states like Minnesota? Do you expect the same priority scheme?
I expect the same priority scheme, yes. Probably the initial doses of vaccine will be distributed on a population basis. In other words, there'll be so many doses for so many thousands of people. So in Minnesota, that has a population, let's say 1/5 that of California, they will get 1/5 the doses initially. I think that's will be true. But within Minnesota, the rules will be the same. Healthcare workers, vulnerable, otherwise capable adults and children.
Can you comment on the effectivity of a state-by-state distribution scheme versus federal plan, or do you expect a change to the distribution scheme under the new political administration?
No, I think everything is going to be pretty much the same. I think people have spent a lot of time working these details out. I think that part of the transition, I think, will go relatively smoothly.
Okay, one question that's asked here. By the way, it is almost 1:30. Do you anticipate that when the vaccine is widely available for all, including children, that this will be added to the required vaccines for school attendance?
Yeah, I would guess this is not going to be required for school attendance. I think right now we have too many other controversies with the virus to get into that. And remember, children who get the illness tend to not get very sick. So children tend to have very mild disease. There are certainly children who have died of this, but they're very few and far between. So I don't think it's going to be a requirement for the school, at least not initially.
And when will phase 2 group most likely be able to get the vaccine in California?
The phase 2 only applies to the period of time when the vaccine is being tried. So I think the phase 2 people have actually probably already gotten the vaccine and already gotten 2 doses.
I think they were talking about the second phase of the actual rollout of the vaccine, the second step.
So, yeah, what do I think? I think you'll see that there will be vaccine released very early on, probably by the end of February, March. I think the people phase 2 start coming in March, April, and then phase 3 probably May, and then phase 4 we'll be giving it to kids who want to get it. That'll be June. That's how I think it will go.
I do need to thank Dr. Cesario for spending time with us again. It's always a wonderful experience when you come because we get to learn new things, and each time you bring new information to us, it really is very beneficial to all of us, especially us who are doing planning for future activities and things. This gives us a little more understanding of what's going on.
Well, good. Thank you. I'm glad to do it.
So I thank you, and I thank all of our attendees also. And I'm going to have to call this meeting to an end. Sorry if we didn't get to everybody, but hopefully doctor will come back in April. Yes, maybe. Huh?
All right, we'll see. Yeah, okay. All right.
Okay. Great.
Okay.
Well, thanks everybody, and that is the conclusion of our presentation for today. Thank you.